outside the John F. Kennedy Hospital in Monrovia, Liberia. 20-year-old Ibaga Stiwo is sick.
Oh, no! Oh, no! Brother, what would you now?
His parents wake beside him. All signs point to the late stages of Ebola, the deadly virus that's killed more than 2,300 across West Africa.
He just vomiting, he toiling, vomiting, toiling, he eat, he vomit. Don't let him get a paper back.
As sick as he is, he won't be allowed inside. The largest hospital in the nation, and many others just like it, are completely full.
Thank you very much. So it's really nice to be here. I'm going to talk to you about Ebola virus emergence. I've packed it full of science. I'm going to try to use the language that I use every day, and I'll give you a few introductions to those. I'm also going to try to manage going between the microphone. I tend to walk and point at the slides a lot, but I think my voice carries. Please tell me if at the back you can't hear something, and we'll make a start. So I actually like to start with my acknowledgements, and a lot of the work that I'm going to show is actually published, so you can find it. Nearly all of the work that I do is open access, so you can find it and read it yourselves. You don't need to be a member of a university or another academic group. Generally there's photo credits on the slides, unless it's my photo. There's references on the slides if you want to go and look up the publication, but there's just a little bit of work that isn't the case, and that these 5 names here are my colleagues and collaborators, and there's some unpublished work which is in it. So this is kind of in real-time science. And I want to acknowledge my funders these days, which is the Royal Society, who fund me as a Rutherford Discovery Fellow. So in the talk, I've divided it into 2 parts. And actually the first part you can really get from Wikipedia, and the second part you can't. And that's what we're doing here in New Zealand. And the first part, I'm going to touch on what an Ebola virus is. What do we know about the origins of the virus? And then I'm going to talk about the West African Ebola virus outbreak itself, which is the biggest one ever. And then why we think these viruses cause such serious disease. And then in the second part, I'm going to talk about how we're trying to understand why and where these viruses emerge. Okay? And I'm going to flip between these. So these aren't distinct categories. So you'll find me going in between them, and hopefully it'll be clear as we go through why. So there's some terms that I'm going to use as well. And I say this because I know I'm guilty of using occasional jargon. And one of them is phylogeny. So by phylogeny, and you're going to see a bit of this, it's family tree. Essentially, it's family tree. You're looking at genealogy. Transmission or infection, I tend to use interchangeably. So transmission of disease, or that is an infection. I also use the term spillover quite a lot. And by spillover is a term we've started to use, and it means cross-species transmission. There's a fantastic book called Spillover by a guy, David Quammen, who's a really good author. So if you read that book, you'll get pretty much what I do. A lot of my friends are in that book. I'm going to use a term called reservoir host. And typically, if I'm saying reservoir host, I'm talking about the population. I'm tending to use that when I'm talking about population, that maintains an infection. So humans are the reservoir host of measles. Cattle are the reservoir host of Mycoplasma bovis. And I'll also use susceptible host. Again, I'm tending in this talk to use it meaning an individual that's susceptible to disease. And hopefully how I'll use these words, it'll become clear. And another thing that I'm aware of that I also use is the word model. And model, I'll use model later a lot. And models predict this and models show that. And you know, we have a conceptual model and then also a model that's a simulation of reality. And essentially, most of the models that I'm talking about here will either be simulations of reality or there'll be statistical models that are trying to link one thing with another. And another term I know I use, and I've used it a few times, is trait. And trait is just characteristics. So one of my traits might be brown eyes, or it might be height, or it might be number of offspring that an animal gives birth to. So that's a trait. So these are just some terms that just sort of introduce them to you, and you'll hear me use those as I go through the talk. So back to the beginning, and really what is an Ebola virus? So this is an Ebola virus. So the phyllo part is like phyllo pastry, or phyllo is like string. And you can see this is electron microscopy of the virus. And it's very simple. So it's only got a few bits. And those bits are the gene itself and essentially some things the gene codes for. And it has a very, very small genome, if you like. So, you know, bacteria's got a few billion nucleotides, and a few million nucleotides, and viruses will just have a few thousand. I want to highlight something that I don't think a lot of people know, and that's that there are a lot of different viruses. So this is the family tree, if you like. So if you just think of your family tree, these are all, and I should have introduced the word filoviruses. So filoviruses taken from that shape, filamentous filoviruses. These are all the different filoviruses, and this is the Ebola one. These ones here. There are these other viruses, and some of those we know have killed people or animals, and some of them haven't. And the colours here go to the map. And I'm going to talk a little bit more about that in terms of, though we tend to think of this as an African problem, and that is true, but we're not too sure what the risk is elsewhere. And we can come back to that. So, when there was the outbreak in West Africa, there was a lot of uncertainty, and a lot of people were looking for information on this, and so you'd have something like CBC News, or in fact BBC, saying things that you should know about Ebola virus. And the reason I had a picture of bats at the beginning is that Ebola virus has been linked to bats. And so at the beginning of the West African Ebola virus outbreak that killed a few thousand people, the Ghanaian government banned the eating of bats in their country. And of course, so I was asked about this, whether it was a good idea, and you'll discover why. I think that probably didn't matter then, is because it was a human infection then. It was humans infecting humans. But the reason that they talked about banning bats is because of the origins of the viruses. So if we think about how you might have, and what knowledge we have about where these viruses come from, who they infect, I'm just going to go through a cartoon that will begin to explain that. So we know from outbreaks in parts of Africa that people infect people with Ebola virus. We think we know that bats are infecting other bats. And we think there's what we call intra and interspecies transmission. So i.e. humans infect humans, but also humans might infect apes. Here it's one bat species might affect another bat species. And there's also then bats infecting their own populations. And we know that people are have got Ebola virus from apes. One of those pictures that I showed up earlier was in the Philippines. We know from work where they've got the complete genome of one of these things that pigs can be infected. And this is all these different Ebola viruses, so it's not one particular one. We also know that duiker, so other mammals, the little forest antelope, have been infected. But we don't know how, or if primates like gorillas are getting their infection from bats. We don't know if bats are directly infecting people. And we don't know what the role of duiker is, what the role of pigs are, if there's other mammals, if there's any transmission back, or if there's any vectors. So actually, to be honest, we don't know a lot. Right? If we did know a lot, we might be able to do more about it, though some things, even if you have a lot of knowledge, are still hard to actually prevent. We know a lot about measles, for example, huge amount, but we still struggle to control measles because it's so infectious. Talking about these different viruses, so this is the distribution of all the species that have ever been linked to Zaire Ebola virus, which is Ebola virus, the one that people think of, that's killed many people. And actually you can see there's some in Asia, but I put a little note under there, not isolated from bat. That's because we've got a lot of smoking guns, but we haven't got the bullet, if you like. We haven't isolated the complete virus from a bat. So there's so many pieces of information. We've got little fragments of RNA, it's an RNA virus, not DNA, but we've got little fragments of the viral genomes. We've got antibodies against it that suggest that these animals have had it, but we can't actually find the virus. It's the needle in the haystack thing. There's another, its cousin on that family tree, Marburg virus. This isolated. There's lots of good evidence this is a bat-borne infection. And it's an African infection so far. But believe it or not, there's a European filovirus, or Ebola virus. And people found it by chance. Never infected anyone as far as we know. Is it because they were using modern genomic techniques to look for whatever's in there? And we can use these next generation sequencing technologies just to discover DNA, and in this case RNA, and they found a complete virus in bats in Spain. But no one's ever been infected with it. Rest of the Ebola virus is the one that they've isolated from pigs, and actually the first links to the Philippines were because they discovered it in a laboratory in America, which is why it's called Reston, Virginia. And people were working in a lab and the primates were dying, and the primates had come from the Philippines. And so, but if you look at the distribution of the bats that have been linked, again this is one where there's lots of little bits of information but not the complete story. If you look at the distribution of those bats, actually it might be all over Asia and Eurasia. So to me that's quite interesting. And it's interesting when you have something, but it's also interesting when you don't have something. So this is what you saw pictures of in the West African Ebola virus outbreak, which is essentially people being carried in body bags because of a highly lethal virus. But there's lots of people living in contact with potential filovirus hosts throughout Asia, and you don't see this picture. Now that could be because we're not looking, and people aren't looking. There's a lot of undiagnosed illness in the world. But it might be, and it might be there's something about the virus. So these are all open questions that we don't really know, but it's still interesting to work out why you might expect something but not see it, as much to say, well, there is something and we're seeing it. So this is one of the tragic cases in West Africa, and you can see how poor these communities are. In the midst of this is early in the West African Ebola virus outbreak in October, so this is October 2014. It actually started in 2013, and there were some misdiagnoses because again people sort of say, well, we don't think it's in West Africa. And if you think about the enormity of this problem, so I'm going to talk a little bit in a moment about what the disease does, but if you look at somewhere in say Sierra Leone, and you look at the size, so this is 300 to 400 cases, and this is in a, I think, 21-day period. So these countries with very poor health infrastructure are getting, just in one city, are getting over 100 cases of hemorrhagic fever, if you like, Ebola virus disease to deal with a week. Now, New Zealand would be crippled by that, you know. I mean, so you've already got an healthcare infrastructure on its knees, and how we then expect them to deal with this. where in the UK or in the US or New Zealand, if someone had this, they'd be put in strict isolation, there'd be huge, basically quarantine procedures in place to prevent that person infecting anyone else. And these guys are having over 100 a week just in one city. So, thinking about these, and we're going to get to, there's a big, been big discussion about how that was or wasn't managed well in the media, and you can read a lot about that. I'm going to talk a bit more about some science related to that. So there's a couple of things. This is again early days of the epidemic, and this is not an epidemic curve. It's just a bar graph showing the cumulative cases through time. And the two things I want to note: this one, which is here in Guinea, which is the blue, and actually this bit here, where these two lines are going up. Ignore Nigeria. This is cumulative cases, so you can see that you've got a few, and then it kind of plateaus, and here then it's going up, and it's starting after the blue. And if we go back to these family trees, so we're talking about Ebola virus. This is all of the Ebola viruses. So going back from some genes, going back from mid-1970s in Democratic Republic of Congo. There's one family tree. These are now disappeared. Democratic Republic of Congo, Gabon, and that top bar is the family tree of Ebola viruses that you're finding in West Africa. And then just zooming in a little bit more, because this is using genetic data. So that, if you think that's the West African part, top in the red, this is scaling in and looking at all the tree. It's not just granddad and grandma and just two examples. It's all the grandkids, it's everybody. And what's interesting, or interesting to me, to us, is if you remember that little blue bar, the first one in Guinea, it just went along and it flattened. That's this first peak. There's a few cases, and it's one little branch of the family tree. And then there's the second peak, which is a bit later, and that relates to this great big bit. And what it does, and what we use this genomic information to do, in fact I'll just go back, it suggests that it nearly went extinct. So it was nearly like all the other Ebola virus outbreaks in Africa, which have sometimes infected 1, 10, 200, 300 people, and then they've died out. And that was probably happening here, and then you got this second big wave. And that's when it hit, and that's when it spread, and it started to spread in time and space, in these very poor communities, bigger urban areas as well. And so it was, and you think, gosh, those few chance events, if only a few things had been done differently, it might have saved thousands and thousands of lives. So anyway, I think it's really interesting, and you can, there's a mixture of epidemiology and genetics and genomics and a whole bunch of stuff that give us this information and say, gosh, a little bit more effort a little earlier on, and you might never have seen all those cases. What I want to tell you, this slide is to remind me to say, you can find a lot of this online. A lot of this outbreak was also really interesting from a scientific point of view, because they got to the point where they were almost doing in-field genomic sequencing, sending the sequences to the UK, and it was almost real-time genome epidemiology. And you can go online, and you can play with these. You can see how the virus is spreading through time and space. And I didn't put any videos in here, but they've got graphs that will show you a video, and you can see how the virus is spreading. And they've not done that because they've known that a person has gone from place to place. It's because they've tracked the gene of the virus. So there's virus A, and there's virus A1's daughter, and there's its granddaughter. And of course it's not, they don't quite replicate like that viruses, but get the picture. And just the sort of exquisite detail that you can see now is that this is Liberia, Sierra Leone, Guinea, and you can see these are all the little chains of transmission in maybe a family where they've isolated the virus. Now, half of that family might be dead, but you can see they're using these chains to try to reconstruct where the virus went and where it went in time. Now, how they use that now, so now the epidemic is definitely finished, but this exquisite detail means, for example, there was some cases some months later, and the question was it a new outbreak or was it something else? And actually what they found was it was dormant virus that had been spread through sexual transmission. And the way they knew that is because they could trace it to the chain, the family tree, and they could say actually it is related to these, and we know there's some other evidence to suggest that came from that person. I mean, that exquisite detail meant that they knew it wasn't a new outbreak. Now, it still needed containing in the same way, but it wasn't from another source. So, but then it poses the question. So I've got it pathogenesis in this, but basically, why do these viruses kill like they kill? And they do kill. And again, you know, this is just for me a shocking example of what people were having to do at that time in these countries, basically bury people in bags, because they were struggling with how to bury the dead in these situations. And actually, I put this as a reminder, so I actually go into this because I've worked with gorillas in Congo. Primates, great apes, as far as we know, are equally susceptible and have died in their thousands in Gabon and Congo. So it affects apes in a similar way. So this is like a timeline of the course of infection. And there's a few things. So time is on the bottom, and you've got your clinical illness. So they're infected, someone is infected here, first week, second week, third, fourth. There's a few things here just to highlight. This on the y-axis, the up going up, is just highlighting the black marks, which are, this is viral RNA in blood, i.e., and ignore what it says on the right, it's the amount of virus that you can detect. And what I think is interesting, and then we've got what the illness is. So the early is incubation, early illness, peak, and then when you recover. So generally about 3-week illness. What I think is interesting, early illness, when people present, if you look by that, roughly around that red arrow, when they present with neuromuscular pain, fever, they've already got lots of virus in them. So already infectious, got a lot of virus with them. And then actually, as they go through this fairly horrendous disease process, actually the virus the amount of virus is decreasing as the body mounts an immune response. And we'll touch on a little bit of that. There's a couple of things. So, for example, the diarrhea. Actually, diarrhea is pretty, in those environments, it's difficult not to infect other people or to manage yourself, especially if you're really unwell and you've got diarrhea. This is a little bit complicated, the slide. We're not going to go through all this. Only I've rather liked it because they've got the bats and they've got everything in this. But this is really showing the process of disease. And there's a few things that I want to highlight. One is thinking about why these viruses are so lethal, and we're going to come to this sort of ultimate and proximate causes, and we're not going to go into too much detail, but things like this, that it infects immune cells. Right? So it's infecting cells that are your immune cells. That's generally not good because it's going to places in your body that it's being distributed through your body, and also you need those. Things like you get secretory diarrhea, so diarrhea is obviously not good if you're thinking about transmission for control. It's not good for you as a patient because you lose fluid as well. And then things like this, the increased vascular permeability. This is where why these things have their hemorrhagic name. So hemorrhagic fever is because a proportion of the patients have hemorrhages. So basically you're bleeding in lots of from different orifices and eyes is one that seems to capture the imagination. It doesn't happen that often, actually. Most of these people get fever, muscular pain, diarrhea, and then go into some sort of DIC, they call it. It's a vasculature collapse and the organs start to fail. But some do bleed. But these kind of things where it's affecting how the water is able to be moved and maintained in different parts of your body is one reason why it causes such bad disease. Another reason why it might be so fatal, I mean, so it is a serious disease, is that this is run in the hospitals. So, you know what, they don't have enough healthcare equipment to deal with routine things. So how are you supposed to keep someone alive and isolated and healthy in this? I think there are, I can't remember if there's 11, 12, 13, 11 cases in the USA, and I think only 2 of those died. That's a much lower case fatality rate than in Africa. And there's lots of things like that. That, you know, children with diarrhea generally do not die in the UK. Actually, they generally don't die in very poor countries, but they much more frequently do. So there's the facilities. This is one, Kenema, Sierra Leone. This is a hospital that was used to dealing with hemorrhagic fevers because they were used to dealing with Lassa fever, which is from rodents. Nearly all the nursing staff died of Ebola in this. So you know, this is how serious this particular outbreak was, and how difficult it was to manage. There are some other things, some other interactions. I put them in there because I think they might be interesting. I don't think this held out. There was one early study that said, well, patients are already infected with this other virus, GBV, they're more likely to survive than those that weren't. And I don't think when this large case numbers came up, that ended up being true. But in terms of lethality and control, this is a really nice figure that was in online. Along the bottom axis, we've got infectiousness. So up here, we've got malaria and measles, which are very, very infectious. And up here, we've got lethality or deadliness. So actually, malaria kills a lot of people, but it doesn't kill that many, given the number of people it infects. Up there, you've got things like rabies, which kills everyone. Basically, if you get rabies, you die. I'm happy to discuss that. And there's Ebola in the pink with a big red arrow. It's not that infectious. It should be controllable. SARS, typhoid, scarlet fever, it's seasonal flu, but it is quite lethal, give all things considered. So, but there's some, for me there's interesting things in that, in terms of it's not that transmissible. And that comes back to why someone like me, who started out as a vet, is some veterinarian by training, working in these. And that's because we think nearly all human infections, with some exceptions, originated in other species. So tuberculosis, probably cattle, but it could be human infected cows, but they're very similar. Measles is very much like rinderpest, which is a cattle disease, now eradicated thanks to vaccination. The overwhelming majority of infections come from something else. We may be infecting other things these days. And Ebola is one here, and actually the point is that it's not very transmissible. And that may be because it's not evolved, it's not adapted to humans yet. So one of the big concerns during that Ebola virus outbreak was it going to adapt to humans and become really transmissible? So, and then have a high case fatality rate, but also be transmissible. And that was even more scary than something that's already quite scary. There's other things, I keep touching on bats, and bats are thought to have adapted to living with viruses for various reasons. I can talk at length about that, but I won't. But in terms of disease, so they control infection, they appear to control infections differently. And one of the reasons, I've just highlighted from one paper, these things, interferon beta, and the little arrows, bats are able to produce it, produce quite a lot of it all the time, but don't produce it in abundance when they're infected. And some of the disease that we see when we're infected with infections is actually our own bodies fighting the infection. And bats are able to kind of keep a low level of that going without causing disease. So sometimes it's our own bodies fighting those, and that's that interaction between your own body and an infection. And things that have adapted, essentially they usually become a bit weaker, if you like. It's not, generally on the whole, it's not good to kill the thing that you need to reproduce. Rabies is another exception again. Rabies is good because it's no good or bad in it, but rabies is able to keep maintaining transmission whilst also killing the thing that is transmitting it. Often that's not the case, because if you're dead, you don't get the chance to go and cover your wife or interact with the kids. Right. That to me, that's all the sort of basic stuff, and breathe, relax. This is the next bit, which is really what we're doing, and particularly what I'm doing here in New Zealand, still trying to understand this and trying to help understand where these things are coming from and why they emerge. And I'm largely going to be talking about this link between bats and people, and the bits in green, sorry, the red-green colourblind, but the other animals in between, I'm going to focus on a bit less. So here, this is the same picture that was on the front slide. So this is a collection of bats. These are Eidolon, straw-coloured fruit bats in Ghana. And actually, all this black stuff on this tree is bat, generally, most of it. Some of it's tree trunk. Most of that is bat. All these dots, these are bats. This is the hospital. And I have seen people on trolleys being wheeled underneath these bats. So we went down to Africa and we take blood and we take it back to the lab and we're looking for antibodies. Interestingly, that on your right, that's that species, and we can find antibodies against other potentially pathogenic viruses, things that kill you, but not Ebola. And yet, this is a species here. Yeah. So this is, if you like, the prevalence, the proportion of the bats, and hardly any of them had antibodies against Ebola. And the one on the left, this is another bat that doesn't roost in these large numbers, does roost in urban areas actually, but and we found quite a few of these had antibodies to Ebola. We're not saying they had Ebola, but it looks to us, given our test, that just like if I took blood from people in this room, you'd have antibodies against flu because you've had flu. You'd have antibodies against measles because you've either had it or you've been vaccinated against it. Most of you, looking at your age, will have had it. I had it. But younger kids these days will have been vaccinated against it. So most of these bats, a lot of these bats, it looks like have had or been exposed to Ebola virus. The question is, why do you get those differences? And there are cellular differences potentially. We don't know if these come first or second. And just to show you, these are the different colours are different bat species across Africa. There's a little bit of different viruses in the middle, and this wasn't our work. The point I want to make is this red arrow. That bat that had the low levels, even if you just take its cells and you try to infect the cells in a laboratory with the virus, they're not very permissive to the virus. They don't allow it to be infected. Whereas other bat cells do. Suggests that another bit of evidence suggests that bats might be the host of these, but not maybe that particular one. There are some maybe ecological reasons. So from Marburg, I talked about Ebola virus' cousin, Marburg. This also causes fatal illness. It's in Rosetta sigilliacus, so it's an Egyptian fruit bat. And there they've shown that birthing. So when the bats have their young, a bit later you find an increase in those young being positive. So a bit like kids going to school, they'll go to school, they'll get infected, they'll come back. It's that seasonality of bringing young, naive or susceptible hosts together, which means that they get infected. So, I left some maths, but don't worry about the math. But in terms of the dynamics, and I'm going to talk about models in a minute, that's affecting the susceptible host. And what we do is we do some maths, and we can simulate those. So it's a bit, what we do is we just simulate the number of in the blue, the young being born each year. And we take some data, different bits of data, and this is the beauty of models, is you can stick it on the computer and go, right, we're going to say that they're infected and infectious for a day or a week, and see if that makes a difference. They're going to give birth once a year, or twice a year. And we can simulate that, and these are the dynamics. So it's a bit like a population of people, and instead of giving birth all the year, they have a big spike of kids. They all get infected. They infect some of the adults, which are the orangey ones, and then a few are still infected, and then there's the next spike of kids going to school, being born. Except these are bats, not kids. And that might tell you something about the risk period, but we're really interested in what does it tell us about persistence in the population. And if you take, we don't need to look at the details on this, but the key thing is, this one is different. Down here this is different, and the difference in this figure is essentially that long line of dots, which means that in those simulations the virus persisted. So just on the computer, not real life, the virus has persisted. And this is using different kind of bat lifestyles and different virus data from experiments. And the interesting thing in that one is that it was the one where we had 2 birth pulses. So maybe, and we know that that other species that lives in cities doesn't have these 2 birth pulses and other ones do. So maybe that is important. And then going to the data, again not showing all the details, but if you split the bats into bats with one giving birth once a year, giving birth twice a year, and you look at the numbers that are testing positive against Ebola virus, then actually the ones which have 2 births a year, there's much more grey, there's much more of the grey charts. And that is the antibodies against it, much more likely to have antibodies. Could be other stuff, but there was some support from the literature. And when you've got a couple of hundred bat species and you've got the whole of sub-Saharan Africa, anything that gives you something to focus on and give you a hypothesis to go and check helps you at this stage. Because if you remember that, the transmission thing with all the question marks, we just don't know. So we have this dynamic model that predicts reproductive rate may be important. And again, it's all may. Other traits, there's that word traits, so other things, height and all the other things. So what we did here is we took, this is just a map of the distributions of all the different bats that we think have been linked to Ebola virus. And we said, what is a bit, you hear these criminal profiling things. So for this we used artificial intelligence, they called it. So we used these artificial intelligence models, boosted regressions, it's got lots of different names. We basically said, we'll give you a little bit of data and say, right, that's what a bat host looks like. And they go, right, has it got 2 wings or has it got 4, 6 or whatever. Bats only do have 2 wings, just to clarify. Which are their hands, by the way. They fly with their hands. Yeah. And then we get basically a profile. And interestingly, these are the top things. You don't need to worry about what they are. I've just highlighted in red the fact that reproduction came up in this other model, this kind of statistical model. And I put this slide up because I think it's really important to show for different reasons. So the top figure is a map of those distributions of all the bats that we think have been linked to these viruses. And this is all of them, all filoviruses, so not just Ebola, all of them. The bottom map is because is a good example of all models are wrong, right? But some models might be useful. So the model can say, it just said, right, bats that look like this, we're going to predict them across all bats because we use all bats. And it says we might have some in America. Now we don't know, but we don't think that those viruses are present in any of the Americas. We could be wrong because there's some little elements of the virus detected in some animal genomes, but we didn't tell the models that they couldn't be there. So it still predicted that they could be there. So the model's wrong, but it's turning out that it's actually been quite good. So I say our 5th ranked one out of well over 1,000 bats was just found positive in Singapore or in Southeast Asia for one of these other viruses. So we think they're useful. They're wrong, but they give you something to go on when you haven't got much. And this is just an example of that. So these are just a whole bunch of new viruses that were found in one of those top-ranked species from the model. And just literally last month, a new virus from Africa, which is in the orange, again in that family tree. This is from Ebola virus from people. These are some of the other ones. And there's a new virus in one of those discovered from one of those bat species. So those kind of things that we're doing in terms of trying to understand what's out there, using these machine learning algorithms, artificial intelligence, whatever you want to call it. The other thing that Ella touched on that is my interest in, and we're still, and it's partly because of this systems like this, is what's happening when we go into these habitats. You know, so I've got this bit in here because people say, well, why not kill the bats? The Ugandan government, when they had a Marburg outbreak, said the Minister for Tourism said, we're going to kill the bats. That's what we're going to do, we're going to kill the bats. And actually, for rabies control in South America, there's a lot of bat killing. So vampire bats spread rabies to livestock in South America. And so what we do is they go in caves and kill all the bats. Often they're not the right bats. Sometimes they are, sometimes not. Yeah, and we haven't got good evidence that stops any of these disease transmission events. So friends have looked at things like the economic importance of bats, and it will be very different for New Zealand. New Zealand will be very different. There are 2 species of living bat right now. But so in the US, bats are estimated to save about 3.7 billion a year from crop pest predation. So, because they basically prevent, because they're the only nighttime insect feeders, really. Basically, birds aren't doing it. So if you think of crop pests, the amount that you're saving in terms of insecticide that has to be used is phenomenal. And that was their lower estimate. Their upper estimate was around 53 billion a year. So, and that was just to agriculture. That wasn't to the forestry industry. That wasn't for other stuff. In the tropics it's much harder, but so to exclude they excluded bats and birds from these kind of agroforestry landscapes. You know, there's a bit of plantation, a bit of forest, cocoa. So what makes cocoa? What makes chocolate? It basically prevented about 1/3 of loss. So the problem is you couldn't exclude bats and birds. You couldn't differentiate between what's the bats and what's the birds. So, but this is the one I do like as well. So that's the blue agave, which you use to make tequila. This is only pollinated by bats. So to get your premium tequila, you need bats. Bat conservationists like this one. But it's a really important thing because a bit like people, right? I mean, most of us are going to. If we're going to get an infection and die, we're probably going to get it from the person sitting next to you tonight, maybe? I don't know. We don't know, I can see here. But at the top, you know, they provide services. But we have to be honest and be aware they also can carry diseases that cause harm. Actually, they've been doing that for millions of years. The question is what's happening now that's changing it. And a lot of our work about that multidisciplinary approach is this kind of One Health thing where we're trying to link human, animal, and environmental health and try to take a holistic approach to health. But it can be quite hard at times. Going back to this, if we're thinking about those transmission pathways from bats to people, so this is some of my pictures. They don't look very good on this, but these are fresh cooked bats in Ghana. These are smoked cooked bats in Ghana. They're harvested by the hundreds of thousands in Ghana alone, southeast Ghana. So if you're thinking about disease transmission, it's just like people working in slaughterhouses much more risk from those than us, right? So these are the people we might want to work with, as well as this isn't really good for bat conservation. But that's also happening elsewhere. So this is in northern India. These are rosette, not rosette. These are another Inictus fruit bat. But all this, see that grey, that dark grey, that's piles of bat bodies. Right? They've been harvested. People go in the caves, like that. So, and you know, like, so clearly, not all these people are dying. Right? Not all these people are dying. And one of the problems with some of the diseases that we work with, or that I work with, is that they're low probability, low occurrence events. They don't happen that often. But when they do happen, the consequences are really high. And they might not be high for society, they might be high for the individual, but sometimes, like that West African Ebola virus outbreak, they're really high. And these are really hard to manage, because people tell you, oh, we've been going there all the time. And then if someone does die, well, actually there's lots of things that cause febrile illnesses. So it's really, anyway, it's very interesting in terms of places to work. And just to show this, that this is in Sulawesi in Indonesia. Different type of bat, these are fruit bats. In terms of those things that are bringing together people as well, deforestation is one, of course. Globally, that's increasing. I think forest areas in the temperate zones are actually maybe stabilising or increasing. But in the tropics, there's deforestation. In terms of bats, there's potential conservation issues, but if we just think about the disease issues, we did some work where we looked at, we took the outbreaks, so this is just a map of Central Africa. The orange triangles are the outbreaks. And we took the forest, we took satellite images, and we basically made a cartoon of it. And we said, right, is it intact forest? Is it perforated? Is it cleared? Is it edge, etc.? And take the satellite image. And then we looked at 2000 and through to 2014. What's the change and what's the correlation with where the outbreaks are? Well, skip over that one. But what we showed is that these outbreaks of Ebola virus are happening in hotspots of forest fragmentation. So it's where the forest is being fragmented. It's not actually where it's being lost. It's one of these things, if you've got intact forest, no disease. No forest, no disease. It's the fragmentation. It says you're fragmenting it, and the process of encroachment. And we've taken I've got last two sort of studies that I'm going to talk about, which are both unpublished work that we've been doing here. So someone's estimated there's about 300,000 mammalian viruses. And this bit, you know, well, any of them could become human infections. We don't know which one. It's a bit of a spin of a roulette wheel. But we're interested in generalisable patterns, and this is again sort of thinking about the science. So a guy in our lab, he's looked at, if you think about Central African forests, so from there you've got Ebola, you've got Zika, you've got HIV/AIDS, you've got malaria, you've got a suite of infections that generally we don't want. We've looked at the edge density, so where the forests are, where people might be having contact. And we've done something where we've looked at the species diversity in those little edges, those fragments. And then we've looked at the population of risk around there. And then I'll say, it's called something a gravity model. It's basically we're connecting those populations by the size of the population. So basically it's saying people in Auckland are more likely to go to somewhere close or somewhere that's big. And the bigger the closer, they're more likely to travel to, so for example, you're more likely to go Wellington to Auckland because you're two big islands close than you are Wellington to Taiana because it's small and it's further. Yeah? And we stick that together and then we make these sort of pandemic risk models of Africa. And this bottom one is the risk, and then we've got areas what we call the intervention sites. And you can put all this together, and this is, I feel like, the final map. And given risk is where we think you're going to get disease emergence. So you can see these hot bits in green and yellow. And received risk is where we think it has spread to. Now this has got nothing about any particular infection. It's just a model. But what's really interesting is, this is Guinea, Sierra Leone and Liberia, and these are the hotspots where Ebola virus emerged. And we've got nothing on information here, we're just saying biodiversity, fragmentation, people. And this is sort of like, you know, I mean, it sort of makes sense in a way, but when you, it's only when you put it together, you go, oh yeah, makes sense. Anyway. And the last bit of work we're doing, so that's David Wilkinson, he's a postdoc in Edinburgh. And then this is by Reid Rannoch, who's my PhD student with me, who's coming to the end of his PhD. If you remember seasonality, I was talking about those birth pulses, seasonality. So what we've done here is we've taken all the Ebola virus isolations, the first detections, and we've got bats, non-bats, but non-people, and then people at the bottom on that side. And the cartoons are just little pictures of where they were. So we've got space and you've got time. We do the same with where bats are given birth. So if you remember that link to birthing, got where bats are given birth, space, time, different types. 3 different types of bats we've called them, but there's hundreds, but we've classified them as 3 because we don't have the data. And actually, because we didn't have the bat data, we had to predict that across time and space. But anyway, so we made predictions of where these different bats were given birth across time and space. And then we predicted outbreak risk. So we use these as statistical models. So we're predicting where people are at risk. But what we think is neat about this, and so other people have done this where they do space and time together, but they've always used other things like rainfall and whatever. We haven't used any rainfall, we've used where animals are giving birth. So we've got biodiversity, but it didn't matter. And what's interesting is that the bat, including bats and where they're giving birth, improved our ability to predict where people were, where cases were. And it improved the model. And so now we've got this risk map, and this is in space, so in space, and this is time by latitude, risk by year, by month of the year. And right now there's an Ebola virus outbreak going on as we speak. There's been 2 in Democratic Republic of Congo this year. One is where the blue was, and the other is where this yellow was. This is Democratic Republic of Congo. So again, you know, it doesn't prove anything, but our model put that as like the 75th or so most likely time and place. So we've got time and place. And this last one was in the 0.1 percentile, top percentile. It's like 99.9%. The model was saying that is a real likely place. If it's going to happen, it's going to happen in that place, but also that month. So this is where we're trying to put it all together. And this is a very little data. So this now, you know, this needs to be proof, gone back, and we need people to go out and work it out. But in terms of where you're going to put your money and when you're going to be looking, we think this is a useful model. So, quick summary. There are actually lots of these different viruses. They're very sporadic outbreaks, but sometimes fatal, and there's lots of different arguments for why that is. We're still not certain, despite all that, talking about bats, we're still not certain about the animal origins, but we're attempting to use these different models to test different hypotheses, and really to try to predict risk. And this last thing, it's not quite last thing, but this other thing, things will change. You know, there might be one single finding and it will say, oh, that was all wrong. We got that wrong. Because we really are, it's a bit of a needle in the haystack. Right, and then my last slide is, you know, I think also we need to think more holistically about ecological integrity and what we're doing, because some of those drivers in those countries are not just locally driven, they're not driven, there's externalities that are putting pressure on those forests. So, anyway, that's it. Thank you.